All right, last but certainly not least, we will be welcoming Alexi Martin to the stage. Alexi is a PhD student in mathematical modeling of infectious diseases, currently working in the disease modeling unit at Swiss TPH. His field of work is in climate and malaria interactions, so a great segue, hopefully.
Thank you very much. Okay, perfect. So indeed, I just said, so my goal is to speak about climate variability and the impact it has on seasonal interventions. So I'm based at CPH and I'm working with open malaria. So the goal was also to develop a bit like open malaria to include climate variables. But this is the what. So today I would like also to speak a bit about the how actually, because we are in the modeling event. So I'd like to actually spend some time actually showing how we actually merge two mathematical models together in order to take the best of each. But first, before I start and I go into the topic, I want to bring you for a little journey, and I want to bring you to Mozambique, more specifically to the district of Xeringoma, so which is a very nice place. Maybe you know it. Very close by, you have like the National Park of Gorongosa, but there are many Anopheles mosquitoes, and some of them are actually carrying malaria. And this is like the incidence curve of malaria in the district of Xeringoma. So maybe I just want to have a closer look at this time series, at this incidence curve. We can see that there is a very strong seasonality in malaria. So really, like every year in March, there is a big peak in the transmission season. But there are also there is also an interannual variability in the malaria. So it means like there is a big difference from one year to another. There is like a difference in the beginning of the transmission season, the duration, and intensity. But actually, why is malaria fluctuating like this? Well, mostly because of this guy, yeah. The Anopheles mosquitoes is a bit picky, and it's actually highly sensitive to climate. So for instance, if we have like rainfall, if we have like temperature, it's really likely to change actually the vector abundance. And all these actually interannual variations of climate drive this interannual variability in malaria. So just to say a couple of things about our model, Open Malaria, that the model actually is developed at STPH and maintained also with the Kids Institute in Perth. So it's actually funded by the EMGF, and it's written in C++, and it's really used for intervention modeling. It has been used for decision making, but as I said before, it's really like, so far it was not accounting for climate, which is something actually to account for. So usually what we do in Open Malaria, we just assume the season to be strictly seasonal. So every year is supposed to be the same. So it's actually not a big issue if you want to deploy a vaccine, for instance, and you want to actually check the efficiency of a vaccine, because you can just run a year without vaccine against a year with a vaccine. And yeah, but it's when we speak about actually seasonal interventions, a bit more tricky. So seasonal interventions like individual spraying or actually bed nets can also be distributed on a regular basis. Also like SMC, seasonal malaria prevention, also like drugs given at different specific time of the year. You will accept that actually like it's easier actually to optimally deploy seasonal interventions if the seasons are considered as actually strictly seasonal. But real-life data is not really usually exactly strictly seasonal. You have a lot of interannual variability, and so the goal is actually to assess how this change in the interannual variations actually how it interferes with the optimal deployment of the seasonal interventions. So the goal is now to go back to Open Malaria and to try to implement this. So really, in short, just so to be sure you're still with me, so the goal is to account for climate in Open Malaria. So really like to improve Open Malaria with this accounting for climate and its induced interannual variations. So just shortly, on Open Malaria, what we do, we have like the EIR, the entomological inoculation rate, so which is really like the number of infectious bites that a human will get. So on at some temporal scale, so like daily or monthly or yearly. And then actually this EIR will force in the model the emergence rate to have the same shape. So if the EIR is actually strictly seasonal, then it means that the emergence rate will be also strictly seasonal. And then so we'll do the malaria metrics like case, incidence, or mortality. So the goal is to adjust the emergence rate according to the climate. And for this, we can actually use a trick. We can use the vectory model, which is another mathematical model for malaria. And we can use actually this vectory to adjust the emergence rate, and then the malaria metrics will follow. So that was a bit the rationale of the study. So what I want to tell you now today is to speak a bit about the vectory model, the open malaria model, and then actually how we can merge them together and how actually we can validate this model, this hybrid model. And really, like, the main objective of today is really to speak about this hybrid model to show actually that it's interesting sometimes to merge mathematical models together. So how it works, the vectory model, so we'll just like take Mozambique, for instance, and we'll just decompose it into many different grid cells. And in each cell, we will actually collect the information about the climate and the surface hydrology. And then we will really like run all of these cells independently. And for each of these cells, so we'll run the model, and then the model looks like this. So we have like 3 different categories. So we'll have like the larvae, the larvae side, the adult mosquito side, and the human side. And it's really like a model which is been computed. So it's really like we will decompose like the larval side into different bins, and we'll really like every bin is supposed to say information about the development stage of the larvae, for instance. So the larvae will hatch from the egg on the left, on the upper left, and then every time there is a probability that they actually die, or they just go to the next development stage of the life. They will just move, and when they reach the last bin, they will just like emerge into an adult mosquito. And when they emerge as an adult mosquito, they will go for a gonotrophic cycle. So they will blood search and then try they will mate on blood search. And every time they find a blood meal, there is a likelihood that actually they find an infected human with malaria, and then they start like a sporogonic cycle. So there is a parasite development that will happen in the mosquito. So there is also like a cycle for the mosquito. And there is also, of course, like a development of the parasite in the human, but it's not very of interest to us, because what we really want to have access to is really like the mosquito's life cycle. We don't really care about the malaria part of the model, basically, because this will be done in open malaria and not in vectory. So what is important to understand is that the larval development stage is actually like the probability to go from one bin to another is really dependent of the breeding site, breeding availability. So it's really like, because we collect this information about the surface hydrology, so we can really like, depending of the number of larvae, we can actually assess the probability of survival and the probability to develop. For instance, like it's also like the development rate of the larvae also depends on water temperature, for instance. So really like the model on the mosquito part is really on the larvae and the mosquitoes are really like quite fine and well described. And the gonotrophic cycles is also like air temperature dependent. So some additional notes on vectory. So it's a model developed by the ICTP, the International Center for Theoretical Physics in Trieste in Italy. It's written in Fortran. And this is the output we get from vectory. So we can really like get the emergence rate. So this is like, I have daily data from 1990 to the present. And it's really like we can access the modeled larvae per square meter and day. Right, so we can really like see, depending on the season, of the climate, or actually this, the larvae, the emergence rate is varying. Now back to open malaria. So really like open malaria is a bit a different way to model the thing. We don't have really like the larvae. We don't model the larvae. So we have like the human part on one side. And what we will do at the beginning, we'll just have the human part of the model. And we'll force an EIR, so an entomological inoculation rate. So we will force malaria to actually enter the human population. We'll do a run like for 90 years, and this will set up the immunity profile of the human population. Then after 90 years, we'll just like add the mosquito part of the model. And then because we have mosquitoes on humans and the humans are infected, the humans will start to infect the mosquito population. And then in return, the mosquito population will also have an EIR to the human population, which is the simulated EIR, really like the EIR produced by the model. Then, because we need to, if the first EIR was actually seasonal, we need to have a simulated EIR, which is also strictly seasonal. So it means that we will use the emergence rate as a way to actually model and to have like this seasonal aspect. And then at the end, we'll just check that the simulated EIR we are producing, that the model is producing, is actually similar to the first EIR that we included as an input for the model. And if everything runs well, we will just cut the input EIR, and the model is supposed to be running as a standalone model. What is good with Open Malaria, it's very flexible, so it's really easy to integrate interventions. We have like a within host dynamics for the human model, and we have detailed health system. But as I said, the problem is that the emergence rate so far is strictly seasonal. So the goal now is actually to use the emergence rate we got from Vectri to modify the emergence rate from Open Malaria. If I just have the two models side by side, I can have the emergence rate here from Vectri and the one from Open Malaria. And what I want to do is I want to force Open Malaria to have the same difference from the mean, basically. I want to have the same seasonality in Open Malaria that the one I have actually in Vectri. And this can be coded as a larval control intervention, even if I don't have larviciding. Here I can just use the fact that in Open Malaria it's easy to code larviciding to force really like the emergence rate to follow these cycles. So the cookbook for this hybrid model, how I run it. So the goal is really like to define a specific location, a study site. We extract the climatic data. Then we run Vectri, the first model, to extract just the baseline EIR, but also mostly the daily emergence rate. And then we input this daily emergence rate into Open Malaria. And we can run Open Malaria with constant EIR. So we force the emergence rate from Vectri into Open Malaria, and then it accounts for climate and its induced interannual variations, which is what we wanted. But why we didn't use just Vectri from the very beginning, it's because now in Open Malaria, it's much easier to deploy interventions and to actually see how these climatic induced interannual variations will impact really like the effect of the interventions. So basically, we created a hybrid model which is accounting for both climate and interventions. Then I want to validate the model, so I decided to take to go back to my district, my Chiringoma district in Mozambique. So I take the grid cell that is in blue as a proxy for the district. So I extract the climate data from this place, the surface hydrology, and then I have this plot at the end. So in red, you see the empirical data that I showed before, and in blue, this is what the model is predicting. So we can see there are still some flows. For instance, like in 2019, there was a very big cyclone, I think, in Mozambique. So this is why very likely the health system was disrupted. So it's not really able to account for this high peak in malaria incidence. But still, what is good is that you can really see that the duration of the transmission season is really well accounted by the hybrid model. So now what's the plan for the future works? The goal is really to do a full calibration of the model. We are planning to use the DHS data, the demographic and health surveys, for really also seeing how it varies from one place to another. If you go in another place in Africa, like in a place where we have 2 rainy seasons and 2 malaria seasons a year. And also actually how this climate, how we can actually model these climate interventions interactions. So as a conclusion, it was a way actually to indeed turn Open Malaria into a climate and intervention-based model. And now it's suited to analyze the impact of seasonal interventions, so the impact of climate and seasonal interventions. And what is also nice, it really accounts for these geographical specificities, because if we want to change the place, within Africa, we want to actually assess the effect of a clinical trial. It's also important to have access to these changes of transmission seasons across the geographical places. And I think what was really nice also to do with this project was actually to take the best out of each model, because Vectri is really, made by climate scientists, so it's very finely made in terms of climate. So it's also interesting to take this part of the model and bring it into Open Malaria. So I thank you very much for your attention. Happy to take any question.
Thank you so much. I do see a hand in the back, but first I just have to note that I think that we have a challenger to Wes Wong for best mosquito sketch in a PowerPoint presentation. He's coming for you.
Thank you so much for a truly lovely presentation. I'm so excited that this work is happening. If I may have 2 questions. The first question is about kind of these more catastrophic weather events that, as we know, are becoming more frequently with climate change. And I thought Mozambique is a very interesting site to use because they did have a massive flooding event, I think sometimes during your time series here. And I was curious how you are approaching that because, as you know, the effect on breeding sites is complicated. There may be more, but there may also be much less if they're all swept out. So I'm curious how you guys are approaching that because I think that's not necessarily something that we have thought a huge amount about. My second question is about temperature dependence of sporogony and how you are thinking or deciding not to think about that because that's also something that we have struggled with in EMOD. Thank you.
Thank you very much for the very nice questions. I think the first one is a bit tricky actually because we know that it's easy to say, okay, if in a natural rainy season, what's the impact of rain? It's one thing to actually try to forecast this, but it's another thing to really like model like these extreme events because we know, for instance, from a more like an entomological perspective that like if we have really like super high rains, it's also like very likely to flush away the mosquitoes larvae. So it's really like also difficult to really assess if this will be beneficial or so there is really like a highly non-linear effect, which is very difficult to model, I think. And especially for instance, like in 2019, we saw like really like this abrupt peak of malaria incidents. I think it's also because of really like the disruption in the health systems, which is also very difficult to actually really model because you don't really know actually what the impact on the mosquito larvae, but also actually what's around. And I think this is like where the model is really like limited somehow. So I'm not sure I'm really like able to provide a very good answer to how we can do it better. But I think what is good is at least in I don't know actually for EMOD, but for malaria we can also like really fine tune the access to the healthcare services. So it's also maybe a way to we can model actually if there is like a cyclone to say okay maybe we can just try to actually understand what the impact of the accessibility to healthcare services on the population. So maybe one thing. I forgot your second question. Sorry.
Temperature dependence of sporogony.
Right. Yeah. So really like what is good is the vector model is really focusing on this. So they really like made the way to really like they use like all these biological experiments to really like understand the nonlinear effect of the temperature on the probability survival of the adult mosquitoes. On this, they just change the basically survival depending of the temperature, which is like also affecting the gonostrict cycle. Does it answer your question? Or okay, I'm very happy to have a talk after. Thank you.
And if I can sneak in just one clarifying question about Vectory itself, acknowledging that this is all very complicated in terms of how to model these massive flood events. In Vectory, what are the primary things that Vectory itself is using in these analyses? Is it just temperature and rainfall, or is it also taking into account these other hydrological factors that we've been talking about today?
No, actually, that is really like what I like with Vectory. It's really like also accounting for all this surface hydrology. So they really like. There is different levels of how deep you can go with the model, but you can really actually model the type of surface, the type of soil. So really like how actually the water is supposed to go into the soil, etc. So what the likelihoods to have like the difference, how actually the water bodies will stay actually after precipitation. So then it's also very likely, very dependent of the mosquito species because we know that, for instance, like Anopheles gambia, they're much more likely to make the best of very small water bodies. Like, so just after the rain, usually you really have like an increase of abundance of Anopheles gambia, while Anopheles funestus, which is a bit more abundant in Mozambique, for instance, are more likely to be very close to the big water bodies, like the permanent water bodies. So there is also like many things to take into account for this. Yeah.
Incredible. Thank you. Further questions? They might not give us coffee for another 15 minutes. So if you wanna fill the time. Yes, I'll look over here.
Thanks. So now you have this nice approach to be able to incorporate interannual variability. If you're doing a project where you're thinking about the future impact of, say, a new intervention, how do you plan to incorporate ideas of interannual variability into the recommendations that you give?
Can you just repeat? Sorry, I'm not sure I exactly followed what you want to mean by this question.
So we don't know exactly what the interannual variability will look like into the future, right? So you may consider different possibilities. How do you think about communicating that if you've been given a question about what do you think is going to be the value of intervention X versus Y implemented in different ways?
So really mean like in the future, or if you want to forecast basically.
Exactly.
I think this is difficult because it's really difficult to really like have this month-to-month internal variability also in the future. So it's really like not the model is really not meant to be like to give an estimation of the incidence like in 20 years or 30 years. I don't know. It's really like the goal. I mean, what we have done so far was really to take the climatic events and the climatic factors of the last 20 years, and then we can model really like knowing actually the climatic factors and trying to do an in silico simulation, trying to say, okay, what if the climate was actually very, how actually, no, sorry, I repeat, I was a bit lost. No, but if we model all the seasons to be exactly the same and basically know if we really like accounted for these interannual variations, or actually are the interventions so useful than we thought actually. There is like, how actually can we overestimated the performance of the interventions? But it's really like the goal is really like to do an in silico simulation saying, okay, this is too difficult to really measure in real life. So what the model can tell us about this changing of regime. So it's really like I think it's difficult to really like just to detail climatic factors in the future, basically. Does that answer your question? Very happy to talk. Thank you.
If I can follow up on that question as well, I know that you mentioned a sort of bigger model calibration ongoing, and in my head, when I read those words, what I assumed is that it might be doing kind of the past version of, Caitlin, what you're perhaps describing, of saying, well, maybe all these intervention-specific calibrations that we've had in the past have actually been soaking up some climate effects that aren't their fault or that they can't take credit for, depending on which direction it's going. And I'm curious if that's accurate in terms of what you're doing next with the calibration. And if so, I mean, there's so many interventions, what would you start with, really, to think about what are the most climate-dependent pieces?
Yeah, that's difficult to really find like the good data set for calibration for this. This is why we actually want to, the goal of so far what I've done is really like just to adapt, because so we could really like follow the different, we just standardize basically the variable. You can see actually if at least the seasons are very well captured. It's really like not about getting the intensity, but at least getting the good starting point of the malaria seasons, which is the main main objective of the project. And it's this is why actually I just like really use the actual data to really calibrate on the fly, basically. So that's not well calibrated in that sense. But the goal now is really like also just to what the future work actually that I want to do actually with the DHS data is also because in the DHS data we get access to like much more fine interventions, like so we can actually account for just the ITNs or even like IRS or actually many also drugs that could have been given to the individuals. So the goal is to look at all this and to actually also challenge the model with like just naive machine learning models to also see if what we are doing here is also meaningful.
Fantastic.
Thank you.
Anyone else before we go? Yes, thank you.
Thank you so much for this interesting talk. Two quick ones. I would like to see a situation whereby you can replicate this study in Nigeria, because I think Nigeria has the highest burden of malaria. Now, is it possible, or what are the what are the mitigations that's why you cannot replicate this study in Nigeria? And then secondly, I hope you can okay, let me just add that one first.
Thank you very much for the question. I can actually, we can definitely like replicate the study for Nigeria, because we don't rely on anything. We can really like just extract the climatic data that we have actually for the entire sub-Saharan Africa. So it's really like just choosing a grid cell, and we can run a model. This for sure. I just took Mozambique because I actually had access to data in Mozambique, so that's purely because of data availability. Then, I don't think we have actually Nigerian data. So if you're willing to do any collaboration, I'm really happy to to talk to you later.
Incredible. Thank you all so much. Let's give a final round of applause to Alexi. It sounds like you have many people who will be coming up to you in the coffee break.